So I'll pass on now to our second speaker today, who's Dr. Geetha, who spent several years in research and development before completing her PhD in microbiology, and is now a postdoc at the Central Research Laboratory in Bangalore, India. And Dr. Geetha brings with her more than 15 years experience in biotechnology, and she's been a key partner in the GPS project. And so now we're going to hear a little bit more about her experiences from this side of things, from the perspective of a lower middle income country. So I'll pass over to you, Geetha.
Thank you, Sam, and thank you, Stephen, for the wonderful talk. So I will now share my screen to continue with the presentation. Yes, yeah. I thank CRE-Seminar series and Murdoch Children's Research Institute for giving me this opportunity to share our experience with the bunch of audience. I hope our experience will be of interest to the audience present over here. So coming to Central Research Laboratory, Kempegowda Institute of Medical Sciences. CRL is situated in Bangalore, which is a capital city of Karnataka state in southern state of India. It's a part of Kempegowda Institute of Medical Sciences and is attached to a 1,000-bed tertiary care medical hospital. CRL was established way back in the year 2008 with an objective to promote research activities in KIMS. CRL is now recognized as National Reference Laboratory for Pneumococcal Vaccine Immunogenicity Evaluation and also a GHRU India unit. We are also an ICMR recognized molecular reference laboratory and COVID-19 testing lab. Apart from these recognitions, the lab is accredited and comply with NABL, which is an ISO 15189 standard, GCLP standard, and GFGP accredited facility. CRL as a research group undertakes socially relevant research projects in the field of infectious diseases and microbial genomics. We have a strong network of over 100 centers across India, consisting of diagnostic centers, medical college, and hospitals. We do provide training and referral services for serotyping of pneumococci from India. So in this presentation, I will take you through a decade-and-a-half journey of CRLs, its transition from a 2-room facility in hospital to a 20,000 square feet infrastructure dedicated to infectious disease research. So when we talk about infectious disease, there comes a question that many important microbes are prevailing in India, which requires our immediate attention. One such organism is pneumococci. Why did we choose pneumococci for the first time for the study? So way back in 2008, when we started our work, pneumococcal infections were the major public health challenge in India. Availability of the Indian data on carriage, disease prevalence, and serotypes were very limited. And moreover, pneumococcal vaccines were not available either in government or in the private sector. Here, there was a pressing need for generating a robust Indian data to inform policymakers on disease prevention strategy. Recognizing these needs, CRL identified pneumococci as the thrust area for the research. Thus, we began our journey with pneumococci with a study acronymed PneumoNet in 2008. This study estimated the incidence rate of IPD in children under 5 years of age. This was a single location study in which we got only 40 isolates by culture method out of 10,000 samples of bloods. IPD incidence rate was very high in children aged 6 months to 12 months, and PCV coverage was around 92%. For conducting this study, we were allotted 2 rooms in Department of Microbiology of KIMS Hospital with no further infrastructure. During the study period, DSL level 2 facility for handling pneumococci and blood culture facility was established. PneumoNet study was a learning process and an eye-opener for us. We were faced with several challenges of low culture positivity, as Stephen highlighted, mainly because of prior antibiotic use and the limitations of the culture methods. And etiological diagnosis was difficult and was taking time. and very few laboratories in India had access to immunological and molecular methods. Here, we realized that there is a high disease burden in India, but laboratory confirmed cases are less, which leads to underestimation of the disease burden. Realizing the needs for estimating the true disease burden, we continued our research with PDOP study covering pan-India. Here, the limitations observed in pneumonet study were addressed by using molecular method. Limitations of the culture methods were overcome by developing a real-time PCR assay platform for detecting pneumococci. Accordingly, the real-time PCR assay platform facility was brought into the laboratory, and challenges of this diffusion methods were addressed by adopting Siemens walkaway machine and automated method for AST testing. And challenges of serotyping, quelling method of serotyping were addressed by introducing PCR-seq typing and microarray methods in the lab. Apart from these, storing and reviving the organism was one of the challenge. For this, we established a separate lyophilization and archiving facility in which we can store the organism for many, many years. Overcoming these challenges in PDOP study, we could get a positivity rate of 30% by real-time PCR method as compared to 7% by culture method. We were also able to identify serotypes from all these culture negative serum samples by adopting PCR-seq typing and microarray methods. PCV13 coverage was 78% in this study. This was a remarkable observations where we saw 92% of serotype coverage in PCV13, where it was a single location study. But in this multicentric study covering across India, the serotype coverage came down to 78%. And the MDR rate was also around 26%, and resistance to penicillin was observed, 11% resistance was observed. Progressing further in our journey, we conducted another milestone study called Hutch project, in which the prevalence, serotype distribution, and antibiogram of Streptococcus pneumoniae in Indian pre- and post-Hutch pilgrims were analyzed. Interestingly, we found an increased incidence of pneumococcal carriage and MDR strains among post-Hutch cohorts. There were also different serotype distribution pattern observed in pre- and post-Hutch cohorts. This study was awarded the best study at Gulf Thoracic Conference, and we were invited to conduct a longitudinal study on Hutch pilgrims in Dubai. After conducting these milestone studies, we looked back at our results and had several questions in our mind. Firstly, do the available PCVs cover the circulating serotypes in India, where we found a region-wise specific distribution and dominance of particular serotypes during our PDOP study, and how to track the emergence and spread of pneumococcal lineages and MDR strains across the country, and what kind of basal data is available for studying the pneumococcal carriage in children and adults in India, and what are the serotypes or the lineages which are unique to the country, region, and season. And finally, do we have any new serotypes of pneumococci which is yet to be discovered? When we sat back and did a brainstorming session, we realized that genomics is the technology which can address to all our queries. We then partnered with a GPS study group, during which 480 isolates were sequenced, and the data, as explained by Stephen, revealed the presence of highly diverse pneumococcal populations circulating in India, with covering as many as 110 GPSC clusters and 54 serotypes. GPSC1 with CC320 and 19F were predominant strains in India. As highlighted by Stephen in his talk, and high-risk clone of GPS10 with CC230 was identified, which covers major vaccine and also non-vaccine serotypes. A high penicillin resistance of 42% was observed during the study, and an MDR strains were about 45%. During this study, we realized the need for implementing microbial genomics in the clinical settings. At the same time, we had a lucky break, and we were selected as the center representing India in a multi-country study of NIHR. This project enabled us to build the capacity for genomic sequencing locally and provided the training. Data infrastructure was built up. Network was strengthened and extended further. During this period, we shifted from our hospital facility to a college facility. A 20,000 square feet building was allotted to us, which was an old animal house. We completely converted this facility to the now existing genomic facility. These are some of the research highlights of the data obtained from the study. We could get significant genomic insights into the MRSA clones, hypervirulent Klebsiella pneumoniae clones, and Salmonella typhi triple mutant clones circulating in India. During the course of the during the course of establishing the genomic facility, we were encountered with several major and minor challenges, and a few of them only I could list out here. Now, as the project was a multinational study, getting the regulatory approval, as Stephen was suggesting, that MTA, Nagoya approvals, HMSC approvals, FCRA approvals, there were a whole lot of approvals which we need to take before the start of the study. So with the help of the state and central government authorities, we could get all these necessary approvals with a rapid turnaround time. Our next major challenge was in getting the custom clearance of equipment and consumables. These consumables had a limited storage time, and also during the transit time, their temperature has to be maintained at a particular set temperature. So getting the custom clearance of these materials were a major challenge for us, which involved a lot of paperwork and production of a lot of official documents. To mitigate this challenge, we identified a reliable CHA agents at the airport and well in advance, and before the shipment arrived, necessary paperwork, duty exemption certificates were obtained for the easy clearance. For the challenges involved in the procurement process, we went through a series of negotiation process trying to include all possible terms and conditions in one contract and made sure that either the equipment or the consumables are covered throughout the entire project period. AMC or warranty period is extended for the complete project period. Further, transfer of funds was one of our major challenge, where we had regulation called Foreign Contribution Regulation Act in India. For getting any foreign funds transferred to India, we have to go through this regulation and we have to get the approval of central government, which is a long drawn process. For addressing this, we approached BIRAC, a Government of India organization, for the complete project management and funds transfer. BIRAC was very supportive in this regard and agreed to take up the project management process and transfer the fund to KIMS at a limited number of time. When we identified the equipment for the purchase and installation, we made sure that the qualified personnel are available locally to support the team in terms of trainings, breakdowns, and availability of the spares, etc. Getting an hands-on training in NGS, wet lab, and also the bioinformatics was another big challenge, which was addressed by a series of training workshops conducted at Sanger Institute and also through the online courses. So we could thus overcome all the challenges established in establishing a facility for molecular genomics at Central Research Laboratory. The facility, what was existing when we shifted from the hospital, was completely renovated and refabricated to a fully equipped genomics laboratory now. Apart from the genomic projects, there are also 4 other major projects ongoing at CRL. An ICMR-sponsored study on etiology of childhood pneumonia in India, where we are serving as a molecular reference laboratory for the project. A BIRAC-sponsored study on pneumococcal vaccine immunogenicity evaluation, where we will be assessing the vaccine immunogenicity by using ELISA and opsonophagocytic assay. And a Sanofi Pasteur study on etiology of acute bacterial meningitis in children. And finally, Pfizer-sponsored study on pneumococcal disease surveillance across different age groups in India. With this, I have now come to last few slides of my talk, where I will take you through the summary of our journey from pneumonet to the current. So our journey with the pneumococci started way back in 2008 from pneumonet project and continued with the PDOX project, HUDGE project, and GPS study. And it gave a way for establishing the genomics facility at CRL. Presently, with NIHR and GHRU project, we have established an exclusive microbial genomics facility in the lab. To summarize, CRL as a research lab focusing on the pneumococci has sailed its way from 2008 and is currently recognized as Pneumococcal Research and Surveillance Center in India. And our journey with pneumococci will continue with new perspective of genomics and future planned projects. These are some of the publications on pneumococci published from Central Research Laboratory. The slide looks very clumsy. I can put only few publication here. So then I would like to conclude my talk with a few snapshots of CRL. CRL is involved actively in conducting various training programs on a regular basis covering different aspects of clinical microbiology, HUNET, bioinformatics, and genomics. This is a snapshot from a ISWP workshop, which is a workshop on Indian surveillance workshop on pneumococci, which is conducted mainly by CRL KIMS to bring together the doctors and the bench workers working in the field of pneumococci under one roof and discuss the strategies, prevention strategies, and the vaccine introduction, etc. These are the few snapshots from HUNET workshop, which we started conducting from 2018. During this period, we had an opportunity to witness the training from Dr. John Stelling himself, who is the main developer of HUNET software. This software particularly helped us to track the AMR resistant strains across the country. These are the snapshots from our genomics workshop, which we conducted in Bangalore. And during our journey, various dignitaries visited us, and few snapshots are shown here. These are our global partners who supported us throughout our journey. Last but not the least, I wish to acknowledge a large number of people who have supported us in this journey of pneumococci. And I thank one and everyone who has given me this opportunity to present this journey with pneumococci. Thank you, everyone.
Thank you so much, Geetha. That's an amazing, some amazing work that you've got there, and I really love that slide with all the different papers. It's so great to see that projects and your collaborations have been so fruitful. So that's fantastic. So before we go to questions, which just a reminder to the audience, you can post your questions in the Q&A, and if we have some time, we'll get to them. But also what we're going to post in the chat is a link to some feedback. So if you have any kind of feedback, please feel free to kind of fill out that little survey, and also a link to subscribe to our kind of newsletter, so you'll get some updates on what's going on with the CRE and kind of other upcoming events, particularly in terms of seminars and training and things like that. So please feel free to kind of sign up to those. So on that note, I think we'll move over to questions. So as I said before, please feel free to post them in the chat, or actually the Q&A box would be easier. But before we do that, we do have a question from Fiona to Dr. Geetha. So does your lab have core funding from the government, or are you completely dependent on grant funding?
Yeah, for this question, we don't have funding from the government, but we have the funding from our organization, that is the Kempegowda Institute of Medical Sciences, and our parent organization, that is Rajya Vokaligara Sangha. And yeah, this is the major fundings, and we also depend on the grant fundings.
Okay, that's great. And Donald also asks, what would you say are the important prerequisites to setting up these global collaborative initiatives? So maybe we'll go to Stephen for that one.
Yeah, that's quite a big question, and I mean the first simplistic answer to that is money. Having plenty of funding helps sort of grease lots of wheels, and obviously, so the Gates Foundation that's a feature of how they work. They've got plenty of money to help make things happen. But I would say on another more personal level, I would say that a prerequisite is your approach. So I think the approach has got to be very open and generous and just generally altruistic. And the two go together really. If you have good secure funding, then it puts you in a better position to be more open and pushing out the benefits to all of the partners. It really is a partnership. You can't. I think I've seen similar things fail in the past where all of the benefit and all of the interest has come to the central organizing institution, and all of the partners don't really benefit. They're just giving away stuff and not really getting anything back, and that always leads to failure. So yeah, I think it's just the general approach has to be right, and funding is integral to that. So if you have if the people in charge are having to chase their own careers and get as much credit on papers, etc., for themselves to keep their own career going, then that doesn't put them in a good position to be altruistic, I would say.
Okay.
Funding.
So Geetha, I guess from the perspective of a lower middle income country, what's something that you wish collaborators from higher income countries understood better, or something? What's something that you would hope that they were able to learn or better understand about the needs from supporting and a collaboration with a lower middle income country?
So I would say that with the high disease burden in lower and middle income countries, so establishing this kind of facility locally is very important. And many a times it might not be possible to send the samples outside any country jurisdiction for that matter. So sending the samples outside the country has to be met with a lot of regulations. This is one of the things which global partners has to understand. So to address this, establishing a genomics facility locally is one of the answer. And for establishing that facility, technical expertise in wet lab as well as bioinformatics, which we are lacking very much. Those are the major requirements. And also, as Stephen said, that the basic requirement of the laboratory setup like power, internet, that cannot be taken care by any of the global collaborators. Only the major concern will be on the technical expertise and provision of the tools, pipeline development that would aid us in establishing and progressing further in our projects.
Okay, excellent. So Stephen, you talked a little bit about GPSC10 and how that particular lineage is obviously highly virulent, and there's some, I believe that there was serotype switching going on there as well. I was wondering, obviously, how that's kind of led to or how that's informed, I guess, in a way, decisions around kind of higher valency vaccines and now introducing 24F into the vaccine, etc. I was wondering if you could comment about, using genomics, of course, as a tool to kind of inform vaccine design. Do you think that this approach with PCV and kind of higher valency PCVs is the answer? Like, obviously there's a lot of work going into kind of next generation pneumococcal vaccines that are kind of independent of the pneumococcal serotype. And so if you're seeing things from GPS, for example, around capsule switching and, lineages that are virulent, as opposed to just being serotype, what do you think about that? Do you think that we should be focusing our attention more on next generation kind of capsule independent vaccines?
Okay, so I am not a vaccinology expert. Immunology is a mystery to me. But my understanding is that actually the polysaccharide vaccines are very good. They do their job very well. So they raise really good responses in infants. So they're very successful in removing those types from carriaging kids. And that is really the strategy that's being put forward. My understanding is that other vaccines are out there and have been tried, so protein vaccines, whole cell vaccines, etc. And it's one of those situations where you have a non-inferiority competition going on, and no one has basically been able to outcompete the polysaccharide conjugate vaccines so far. The question of increasing valency, it has increased. So it started off with a 7. and over the last 20 years, it's gone up to 20. So there's now the next vaccine that's going to be coming online is a PCV20. So that is a technical manufacture challenge, as far as I understand it. We were very lucky, I think. So in terms of our project being able to actually influence the formulation of that vaccine, I think we were it's mostly explained by the fact that our project is funded by Gates, and Gates are also backing the development of the vaccine. And so essentially Keith Klugman was aware of what we were doing. And so when he's in meetings about the vaccine formulation, that's how it feeds through. But I don't find that very satisfying. I think that we need to have a clearer way of letting the companies, the development companies, know what the interesting information is. And we're working on that. Yeah, so I think we're probably going to be in a situation where genomics can actually be useful going forwards, and we might be able to say, okay, let's look at the current formulation. Are there any serotypes in there that we don't need anymore? And we can drop those and stay under that. Maybe if there's a ceiling of 20, then we can stay within that. So yeah, so it is. I think that, the limitations there are down to manufacture, and I actually, at one of the meetings that Geetha, I'm going to go off at a tangent here. One of the meetings that Geetha showed a picture of, I met Kim Mulholland there, and he was explaining to me how PCV7 first came about and why it was a 7-valent vaccine. Despite the trials having been done on a 9-valent vaccine, the company that was up to make the vaccine only had 7 fermenters.
Oh.
So two serotypes had to be dropped. Unfortunately, those were the ones that were most important not in America.
Oh, there you go. That's a fun fact. I didn't know that. Good to know.
Yeah.
So I might open this question up to both of you, actually. So maybe we'll go to Geetha first. But I was wondering about, I guess, just generally in terms of the overview that you've both given in terms of your work today in the GPS. What are the lessons that you've learned from your experiences going through this whole, this big, this massive undertaking? What would you do differently if you had your time again? Geetha, did you want to maybe go first?
Yes, so getting involved in the GPS study was one of the great opportunity which we have ever got. So with the availability of 480 genomic data, it is like a gold mine. We can keep on extracting whatever the kind of genes information, whatever we require, the expression mechanisms, the complete transcription regulation mechanism, whatever we wanted, we could have mined it. So it was like a real gold mine for us, and we could analyze the circulating pneumococcal lineages, which was tracked by using the GPSC cluster. So that was a good correlation of the GPSC linkages, what we have made. So from India, we had several new pneumococcal lineages which were existing. And in this study, we also came up with the new serotypes. Only some 2 new serotypes of pneumococci were identified. So that was the amazing thing which I could see from this GPS study. Not only this, we still have to extract more and more out of this study. So that was a better learning process. And during this course of period, we were also got exposed to the wet lab training and also the bioinformatics aspects of handling the pneumococcal genomes, and many more amazing things happened. So it was a great learning process for us.
Excellent. Stephen?
That's a really hard question. What would you do differently? Because it's been a long time, and things have changed over time. There's things that I regret: not getting enough funding for bureaucracy, paperwork, etc. We essentially had, in GPS1, we essentially had 2 postdocs who were dealing with MTAs and RCAs and all that sort of stuff, which shouldn't really have been happening. They should have been getting on with science. So maybe being smarter about funding resources for some of the less scientific parts, I think.
Okay, excellent. And we do have now some questions coming in right at the end. So There's one here from Casey. So we've had many different methods were used in the GPS study to find potential teams to include in the study network. So which method did you find to be the most useful to find or build partnerships, Stephen?
I wouldn't say any method was bad. I think, you know, personal relationships are important. So actually getting to meet people face to face is really important. In fact, I remember so Geetha's boss Ravi, they were over in the UK for a conference and sent me an email, and we met at one of the London train stations for a chat. That's where it all began. We got, you know, we hit it off straight away. Had really good relationships there. But I would say, you know, I mean, we keep an eye on the literature to identify people in countries that we're not already engaged with who are writing about relevant stuff. Yeah, it's all useful.
Excellent. So we might wrap up with Geetha with just one last question, a bit of a big picture question, I guess. Given everything that you've kind of gotten out of this, you know, this collaboration and all the opportunities it's provided for you and your team, you know, what advice would you give kind of, we've got many early career researchers tuning into this seminar today. So, you know, what's the advice that you would give them in terms of pursuing a career in research? Particularly from a low and middle income perspective, as many of our attendees are from low and middle income countries. So if you could comment, that would be great.
Yeah, true. So being a basic researcher, so I was not really into this genomics research at all. So I was a veterinary graduate dealing with the dairy microbiology. So later, with my interest, I took up the project in molecular biology during my post-graduation, and then later on continued to do the PhD program. Yeah, of course, definitely genomics, biotechnology, and molecular biology all are like almost similar fields, which offers a great opportunity. Particularly if you are interested in the research work, definitely it's an amazing field. You have lots and lots to learn, and collaboration, you'll get to meet people and know the knowledge is everywhere, so we can gain the knowledge. A lot of things can happen, but unless the people show their interest in research, because in India I could see very limited number of people who continue to stick on with the research, because with the research in India there are other limitations. If other limitations while we face while we perform the research programs, lack of funds, as we say, and a lack of guidance, proper guidance in the field of genomics, bioinformatics as such, and the facility, mainly the facility which will be lacking. So these are the things which makes us to bog down saying that no, we will not do research work. But yeah, sticking on to research definitely helps.
Okay, excellent. There's some wise words there for our early career researchers to focus on. And so on that note, I'll draw things to a close. So thank you once again, Stephen and Geetha, for taking the time and presenting some your work to our CRE and all our other guests from many low and middle income countries across the world. And thank you everyone for joining us today. And we hope that you got something out of it. And please, again, feel free to fill in the feedback on the link there. And also subscribe if you want to hear some more about kind of updates and upcoming things from our CRE. Thank you.
Thanks very much.
Thank you. Thank you very much.